Hi, welcome to Once Upon a Neuron. The next 8 minutes, we're going to take you on a neurological journey that you will never forget, studying how experimental hypothyroidism delays field excitatory postsynaptic potentials and disrupts hippocampal long-term potentiation, indigenous gyrus of hippocampal formation, and Y-maze performance in adult rats. So if that doesn't get you going, I don't know what will.
I'm sorry, what was that? Could you repeat the title, please? The background information. Hypothyroidism is when the thyroid gland doesn't produce enough of the thyroid hormone. So why is that a bad thing? First of all, it can lead to heart problems. You can have an abnormal heart rate or an elevated heart rate. It can also lead to mental health issues such as depression. Along with that, it can lead to infertility and additional birth defects later down the line. The dentate gyrus of the hippocampus is an area that assists in the formation of episodic memory. Episodic memory are things that are chronological, such as when, what, and where happened at a certain moment. One of the more interesting parts about the dentate gyrus is it is one of the select few structures of the brain in which there's a high rate of neurogenesis.
So, Zach, why are these things important?
Well, thanks for asking. Manipulations in thyroid hormones have been shown to influence your learning and memory, and I don't need any help getting the grades I already achieved.
Okay, thank you. So, thyroid hormone deficiency, even of short duration, may lead to irreversible brain damage, and the consequences of which depend on the specific timing of onset and duration of deficiency.
Now we're going to do a question from you, our viewers.
Whoa, live question.
Wow, what do we have?
An EPSP, excitatory
Postsynaptic potential. Look at that.
Great. An EPSP is a temporary depolarization of the postsynaptic membrane. Although the EPSP from a single neuron is rather small, when recorded from an extracellular component, it can easily be recorded with a field electrode in some areas of the brain, such as the hippocampus, when the neurons are in the same orientation, which is also known as the field EPSP. And what does that have to do with LTP?
LTP, known as long-term potentiation, is typically studied using continuous high-frequency neuronal firing, NAPSP. Because of the continuous neuronal firing, the neurons create either new associations between each other or strengthen existing ones, forming memories. So Zach, how was this experiment performed?
We used 12 male Wistar rats, aged 6 to 7 months. They weighed between 280 and 342 grams. They were given PTU, which is an anti-thyroid drug. It reduces thyroid hormone levels. Put in their water for 21 days at the concentration of 0.05.
Oh, sounds interesting. Continue.
So, Sergey, how did we determine the thyroid hormone levels?
Well, you see, Zach, we took blood samples from the hypothyroid in the control rats and not the control rats just after the completion of the recording. We assayed the blood samples at negative 20 degrees Celsius using the enzyme-linked immunosorbent assay kit, and we measured the amount of serum T4 in the actual serum.
Katie, what are some traditional ways that other experimenters have tested some experiments like ours?
Well, originally and normally, the Morris water maze test represents an artificial situation, and which is unfortunately relatively stressful for rats, and we really wanted to keep that high level of stress down. So instead, we did a Y maze, which is, Sergey, you want to tell us what that
A Y
is?
maze, yes, I do. A Y maze is a test where you have a Y-shaped area with 3 terminals, and there are no distinct cues on any of the ends as to which terminal is which, and so it has rather light stress for the rats, and the test is based on the innate curiosity of the rat itself. It will want to go and explore every single section. So how the test was performed was the rat was first introduced to the environment. It became familiarized with the environment, after which it was taken out for one of the trials. It was taken out for 5 minutes, then reintroduced to see how well it remembered the general location. It was always introduced the same way. The sides were randomized for each trial, so some rats at the left side be the place where they started, some rats at the right side be, but every time that they were reintroduced, it was the same spot. And so short term was 5 minutes, then it was reintroduced, and the long term was 4 hours, and then the rats were reintroduced.
So, Zach, do you mind telling us how we verified the electrodes?
Yeah, so we cut up through their stomach after we put them to sleep and reached their aorta, snipped it, and then pumped saline solution and paraformaldehyde 10% through, and we also post-fixed the brains after we took the brains out of the animals and cross-section for the hippocampus, and then we stained it and checked its analysis through fluorescence microscopy using an LSM 710 microscope. So Sergey, how was LTP induced and tested using our Y maze?
I am so glad you asked.
Thanks. I'm
After a
glad you're glad.
15-minute baseline of recording FEPSPs from the rats after they went through the maze and looked at it, there were 4 sets of high-frequency stimuli delivered to the rat at 5-minute intervals, at 15 minutes, 20 minutes, 25 minutes, and 30 minutes to induce LTP. Afterwards, it was repeated every 30 seconds for the next 90 minutes, so that's to induce the LTP in the rats. So Zach,
Yeah.
how could we refine our experiment?
Well, we think our experiment was great, but we know that there's always room for improvement. So if we were going to change some things, one, we would increase our testing size. We wouldn't have 6 animals in each group. We would actually add more groups and have varying concentrations. So instead of giving them 0.05 or 5% in their water, we would be giving them varying concentrations to see the effects that would cause. And also, we would have a female testing group. So these were all males. So we're going to have a female testing group just to see the effects of their development according to their gender. So we know that you, our viewers, are on the edge of your seats dying for our results to be released. So here we go.
So for the control rats, those not injected with PTU, they went to the new arm more often than they went to the familiar arm because they were learning about the new arm as opposed to the familiar arm, which they already were familiarized with. In hypothyroid injected or in rats injected with PTU in their hypothyroid, they went to both areas of the Y maze in equal amounts, indicating that there was no learning going on and both were not familiar for them. So for long term, there was no difference in either of the rat groups. So what this means is, since the control group has already looked at both sides of the maze, neither of the sides are no longer interested in them, as opposed to those injected with PTU, they still haven't learned either side, so the reasoning is they visit both sides continuously in short term and long term, whereas the control group rats visit the new arm more in short term and visit both the same amount in long term. As you can clearly see, the rats injected with the PTU had clear memory impairment, and so this is supported by previous research indicating that thyroid levels do indeed affect the memory in adult animals.
Hey guys, thank you so much for watching our channel. Please subscribe below. Yeah, thank you so much. Okay. Bye.